Agilent Technologies has positioned itself as an industry leader, combining a more than 33,000 square foot state-of-the-art multi-product facility with world-class expertise.
The employees are definitely dedicated to the field of nucleic acid therapeutics and therefore dedicated to improving the lives of the patients, which ultimately will be benefiting from the manufacture of our products.
Set in Boulder, Colorado, a center for nucleic acid therapeutics-based biotechs and with strong ties to the University of Colorado, Agilent Technologies has assembled a world-class team,
which allows us to take on the tough projects out there and deliver them in a timely fashion to our customers. We believe that working with Agilent reduces the risk of your projects based upon the experience that we have and the fact that we have such a breadth and depth of experience in manufacturing products of this kind.
Agilent's team has the ability to take on your projects, whether they are based on established processes or cutting-edge technologies developed in-house by Agilent.
We currently manufacture all of the classes of APIs on this site and have significant experience in the more complex RNA-based therapeutics.
Agilent offers a full range of turnkey development services, including analytical method development, chemical development, technology transfer, process optimization, scale-up and validation, API and drug product stability, as well as supply chain management. With a wide range of manufacturing assets, Agilent can match your project needs to the scale of manufacturing required.
Our total current manufacturing capacity consists of three major manufacturing areas: our small, medium, and most recently large-scale production, giving us the ability to manufacture from early clinical toxicology batches in the grams to hundreds of kilos at commercial.
Agilent's small to mid-scale facility is designed to produce multiple APIs following good manufacturing practices. The facility is comprised of 9 manufacturing suites, covering the entire production process from synthesis through lyophilization. There are 3 synthesis suites, supported by an integrated solvent delivery system. Currently installed within these suites, 2 ACTA oligopilots and 3 oligopilot 400s, with cleavage and deprotection equivalent to the synthesis scale. Following synthesis, downstream processing is conducted in 3 separate suites, consisting of liquid chromatography and ultrafiltration skids. Buffers are prepared in a centralized location and distributed throughout the facility. With flow rates from 200 mils to 4 liters a minute, Agilent can purify grams to kilograms, dependent on the project need. Varying scales of ultrafiltration equipment are available to match chromatography output. An additional processing area is available for post-purification steps such as pegylation, other conjugation chemistries, and duplex annealing. Reactor sizes are available for various manufacturing scales. For final processing and packaging, freeze drying is conducted in 2 dedicated lyophilization suites. Bulk lyophilization is performed in LyoGuard trays in batch sizes up to 5 kilos, with vial lyophilization also available. All process and final product release testing is carried out in-house. As a world leader in measurement and testing, Agilent is well equipped with state-of-the-art analytical instrumentation, including high-resolution mass spec, ICP mass spec, and high-field NMR. Process development laboratories with scaled-down versions of manufacturing equipment support the commercial operation. Expertise in development of the most complex DNA and RNA molecules sets Agilent apart. Agilent's large-scale capacity is the result of an expansion project undertaken in 2008 to meet the demands of our current and potential partners.
We have constructed and installed both plant and equipment capable of now supporting late-stage clinical trials and commercial launch of nucleic acid APIs. This has resulted in hundreds of kilos of annual capacity in our facility. The facility project was completed in record time, partly due to the acquisition of Dow's nucleic acid medicines business early in 2008. With the acquisition of the Dow assets, we were able to rapidly design a facility around those assets and install the equipment within 1 year. We can now manufacture from the small scale, gram quantities, up to kilos. This increase in scale will drive efficiency and drive down cost of goods over time.
The heart of the large-scale facility is the oligo process synthesizer, capable of batch sizes up to 1 mole. This synthesizer and its associated supply vessels are housed in a dedicated 2-story suite. Critical raw materials are delivered to the synthesizer via gravity feed from above. Amidites are weighed in individual isolated feed pots, then transferred to their makeup vessels. Dry acetonitrile is delivered volumetrically to the makeup vessels where the amidites are dissolved by agitation. Activator and oxidation or thiolation solutions are made and delivered in a similar way. Additional synthesis reagents are fed from the internal and external tank farms. Cleavage and deprotection of the oligo from the solid support is performed on column, utilizing an automated post-processing module. RNA desilylation is carried out in a controlled reactor.
We have put a lot of thought and effort into the design and construction of our facility in order to ensure a world-class manufacturing facility capable of both manufacturing the product and withstanding regulatory inspection.
Purification of the deprotected oligo is carried out in a separate suite featuring a 30-liter per minute HPLC. A high-capacity ultrafiltration system with a 750-liter hold tank and 160 square feet of membrane surface area are used for concentration and diafiltration. Further volume reduction is carried out in a vacuum concentrator. Final lyophilization occurs in a dedicated suite. With 36 liters of total volume, up to 5 kilograms of oligo can be dried in a single lot.
We're very proud of the facility that we have designed and built here in Colorado that is now capable of serving the entire nucleic acid therapeutic industry. We have built the facility in record time and are capable of making from the simplest to the most complex molecules that the market requires.
Agilent invites you to see this world-class nucleic acids manufacturing facility, to see firsthand how we can rapidly develop, scale, and manufacture your clinical or commercial API. Agilent Technologies. Expertise, capacity, innovation,